Okay, welcome to Chapter 28. This is our discussion of the reproductive system. And honestly, these last two chapters on reproduction and human development, I think, should really come at the beginning of any anatomy and physiology course because this tells you a lot about um, where the structures we've been studying over the past 30 weeks really come from, why they form, and if you can understand their origins, it's easier in many respects to understand their function. So, what is the purpose of the reproductive system? And it's obviously to perpetuate the species. The gametes are the specialized cells that come together in the act of fertilization to produce a new individual. There are specialized organs in the reproductive system that generate the gametes: the testes, which produce the sperm cells in the male, and the ovaries, which produce the egg in the female. The reproductive system enhances the fertilization of the oocyte by the sperm. The duct system in males nourishes the sperm cells until they're mature and are deposited in the female reproductive tract by the penis, and the female reproductive system receives the sperm cells from the male and transport them to the fertilization site. What else does it accomplish? Well, it allows the development and nourishment of a new individual. The female reproductive system houses the developing embryo and fetus for the 9 months of gestation. Also, during that period of time, the mammary glands begin production of milk in preparation for the delivery of the baby. Um, what we have to understand and appreciate about this is that this represents internal fertilization and development, which is a major evolutionary step forward in terms of investment in time and energy towards producing offspring. Um, because of internal fertilization and development, it's much more likely that the fertilized egg, the zygote, is going to um, manage to make it to neonatality and form a new individual, as opposed to external fertilization and development, where, of course, um, the hatchling would be at much greater risk. As an example, if we were to compare um, external fertilization and development um, to the human system, we could look, for instance, the way amphibians reproduce, which is basically the female laying a clutch of eggs by attaching it to a substratum. The male swims over the clutch and dumps his payload of sperm, and then we generate many hatchlings, many tadpoles, but the likelihood um, that any one tadpole is going to make it all the way to adulthood is minimized because they're essentially at risk as a result of being out in the environment very early on in their development. Most of those tadpoles are going to end up becoming fish food, and very few, percentage-wise, are going to make it to the adult organism, the frog or the salamander or what have you. So internal fertilization development, a major step forward in evolution. What else does the reproductive system do? Well, it generates the hormones that regulate the system. The hormones that are produced by the reproductive system control its development as well as the development of a gender-specific body form. These hormones are also important for the normal function of the reproductive system and reproductive behavior. If we look at the male external anatomy, reproductive anatomy, um, visible is the scrotum, which is a two-chambered sac that contains the testes, which is separated by the perineal raphe. Okay, you can see here the raphe running essentially from the anus all the way to the base of the scrotal sac, and also bifurcating here the scrotum. The dartos and cremaster muscles regulate the temperature of the testes. When temperatures rise in the environment, the testes will tend to drop away from the body in order to cool them. As the temperatures outside cool, the testes tend to rise up closer to the body in order to warm the testes. The purpose, again, is to maintain the optimal temperature for sperm production so that the male has maximum fertility. The perineum is the diamond-shaped area between the thighs. Again, this transcribed area represents the perineum. It's divided into an anterior urogenital triangle, which contains the penis and scrotum, as well as a posterior anal triangle. What you're looking at here is a cross-section through the male reproductive anatomy, and what you should appreciate is the fact that the gonads in the male, the testes, hang outside the body, again, to maintain a temperature between 2 and 3 degrees below normal in order to optimize sperm production. Sperm produced in the testes eventually find their way to the epididymis, this coiled structure here. They get there via the seminiferous tubules in the reed testis, and in the epididymis they become capable of motility. They're able to use their flagella in order to take the long journey from the tip of the penis until ultimately, hopefully, they reach the egg. The vas deferens is the tube that connects the epididymis ultimately to the prostatic urethra via the ejaculatory duct. The urethra in the male, remember, is a common passageway both for urine and for semen. Semen has the right of way of urine during ejaculation. Smooth muscle closes off the urine that would otherwise penetrate the prostatic urethra, allowing the ejaculate to move from the prostatic urethra through the membranous urethra and into the spongy urethra, and eventually out the tip of the penis into the female reproductive tract. And at that point, what happens is that the majority of the sperm actually fail to make it to the egg because, essentially, for lack of a better description, sperm are stupid. They go in any direction that they're pointed. Many of them crash and burn into the uterine wall immediately upon their entry through the cervical canal into the internal os, into the body of the uterus. Of those that don't crash and burn right away, cervical mucus can help guide the sperm ultimately to the opening of the fallopian tubes. Unfortunately, about 50% of those sperm are going to swim up the wrong fallopian tube, missing the egg. And of those that do swim up the proper fallopian tube, many of them will overshoot the egg. Some of them will not make it to the egg. So only a fraction of those sperm are actually going to attach to the ovulated female gamete, and only of those, one sperm is generally going to make it to the egg cell membrane and affect fertilization. Okay, so this is one of the reasons why, if you have a sperm count that's abnormally low, you can be described as what we call functionally sterile, because you need a certain number of sperm in order to overcome these barriers to fertilization that ultimately 86 most of the male gametes. Other structures that are clearly visible include the structures that contribute to the ejaculate. Okay, you can see, of course, the testes, which is where the sperm are produced. As we're going to find out, a male is fertile from puberty until death, as opposed to the female, who is fertile from menarche to menopause. Other glands that contribute to the ejaculate include the bulbourethral gland, so named for its position near the bulb and the urethra of the penis, and the prostate gland, along with, of course, the seminal vesicle. Okay, so 4 glands in total have contributions to the ejaculate. Notice again that the male urethra has 3 parts: the prostatic, the membranous, and the spongy urethra. And we've talked a bit in previous chapters about the fact that occasionally the prostate can enlarge, especially in older men, and occlude the prostatic urethra, and this results in an inability to pass urine. It also can result in an increase in pressure in the bladder, which can back up into the ureters and damage the kidney. So one of the things that we do in this event is that we can either core out this portion of the urethra in order to restore flow, or we can use drugs that will shrink the prostate and hopefully restore flow as well. In the penis, you can see 3 columns of erectile tissue. This is the corpora cavernosa. There's 2 of these paired dorsally on the penis, and the corpora spongiosum, which encircles the penile urethra, sometimes also called the spongy urethra. When a male has an erection, what happens, as we're going to find out in a bit, is that the rate of blood flow into these columns of tissue is greater than the rate of blood flow out, and the result is an erection. The glands of the penis, remember, is covered by the prepuce or foreskin, which is often removed at birth in an operation known as a circumcision for hygienic purposes, because bacteria often live in the crevice between the glands and the prepuce. Notice also that there is tight proximity between the prostate gland and the rectum and anus. One of the ways that we can check for prostatitis or potentially prostate cancer is to palpate the prostate through the rectum and anus, feeling for enlargement or feeling for unusual masses. We often perform this test in combination with a PSA test in order to detect cancer in the prostate. Remember that one of the reasons we worry about cancer in the reproductive organs is because of the proximity to the inguinal lymph nodes and capillaries, allowing a path for metastatic tissue to move throughout the body. The testes are located outside the body cavity because sperm require lower than body temperature for development. The testes are also exocrine glands. They generate sperm cells as part of the seminal secretion, but they also generate testosterone, which modulates male secondary sex characteristics and also has an effect on the rate of sperm production. The covering of the testis consists of the tunica vaginalis, which was originally peritoneal tissue. It is a serous membrane. Deep to that is the tunica albuginea, which is Latin for white jacket. It is a thick white connective tissue that penetrates the testis and divides it into lobules containing seminiferous tubules and Leydig cells that secrete the testosterone. The tubules in which the sperm move are the seminiferous tubules. That's the site of sperm production. The tubula recti and rete testis are the pathway to the epididymis. So you can see here these coiled structures are constantly generating sperm, again, from puberty to death in the typical male. So you can see here from seminiferous tubule to rete testis to epididymis, and then ultimately to the vas deferens, and from there, of course, to the urethra. The testes do not begin life outside the body. They actually are inside the peritoneal cavity. Their descent allows them to pass through the inguinal canal into the scrotum, guided by the gubernaculum, which is essentially a combination of connective tissue and muscle that eventually will slide the testis through the inguinal canal, passing through the inguinal rings, which are openings in the aponeurosis of the transverse abdominis and the internal-external oblique. Remember, guys especially, this is a weak spot in the abdominal wall. Every male has these, and one of the dangers is, as a result of this structural feature of the male abdominal pelvic region, is that if we were to lift a heavy object in an improper fashion by bending at the waist and straining, the result in some cases is to push organs in the abdominal pelvic cavity into the scrotum, and this is what we call inguinal hernia. The danger, of course, is that we can essentially cut off blood supply to that part of the organ. It can infarct and die. Sometimes a fluid-filled cavity called a hydrocele can be forced into the testes as well as a result, again, of improper lifting technique. Okay, so something to be aware of for the male. Females also have these inguinal rings, but they're much smaller than in the male, and the risk of an inguinal hernia is far less in general. Cryptorchidism is a failure of the testes to descend into the scrotum. It prevents normal sperm development, happens in a tiny percentage of male children. There are surgical techniques we can use to rescue the hidden testes and guide it eventually into the scrotal sac. So what you're looking at here is just a diagram showing you how the testes descend. If you look in the upper left-hand corner of this figure, what we want to appreciate is that each testis forms as a retroperitoneal structure near the kidney. They begin beneath the parietal peritoneum, descending towards the inguinal canal. The process vaginalis extends into the inguinal canal alongside the gubernaculum. The testis then follows the process vaginalis and descends through the inguinal canal into the scrotum. Between birth and adulthood, the process vaginalis disappears and its inferior portion becomes the tunica vaginalis, which is, of course, superficial to the tunica albuginea. And then the gubernaculum transforms into the scrotal ligament. It's important that the testes retain their orientation inside the scrotal sac. If they were to twist, there is the danger, of course, that we could cut off blood flow to the organ and it could infarct and die. Okay. Sperm are produced in the seminiferous tubules. At puberty, gonadotropin-releasing hormone increases the production of luteinizing and follicle-stimulating hormone from the anterior pituitary. These levels promote sperm formation. The follicle-stimulating hormone levels promote the interstitial cells to produce testosterone, and this, of course, accelerates spermatogenesis. The spermatogonia divide to form the primary spermatocytes and the daughter spermatogonia. As with all stem cell populations, remember that one participant in the cell division will remain behind as a stem cell, while the other product of division will eventually differentiate ultimately into sperm cells. The primary spermatocytes engage in the first division of meiosis to form secondary spermatocytes, which divide to form the spermatids, which develop an acrosome and a flagellum in the process of differentiation. The Sertoli cells nourish the sperm and they form a barrier between the blood supply and the sperm cells that are produced by meiosis. The purpose is to protect these gametes from attack by the immune system, because you have to remember that sperm are genetically unique from the male that produces them due to the reshuffling of genes that occurs during prophase meiosis 1. So we have to take special care to lock out the B and T cells. Interstitial cells produce testosterone, while the sustentacular cells convert it to DHT and estrogen. They're probably active hormones that cause the sperm formation. As a sidebar, it's DHT that's also thought to be causative agent in male pattern baldness, alopecia. What you're looking at here is just the head, the midpiece, and the tail of a mature sperm cell. And you can also see a cross-section up here through a seminiferous tubule. And what you're seeing going from the superficial to the deeper aspects of the cross-section is a developmental sequence going from spermatogonia, then to primary, then secondary spermatocyte, then ultimately to spermatid, and then finally to spermatozoa. And you can see the flagella projecting into the lumen of the tube here. Remember that this is a process that goes on in males from puberty until death. So let's take a look at how meiosis works.
Meiosis is the process that results in the formation of sperm cells and egg cells. The cells that will undergo meiosis are typically found in the testes and ovaries of males and females, respectively. These germline cells are diploid, having 2 sets of chromosomes, but will undergo meiosis to produce haploid cells, having 1 set of chromosomes. During fertilization, these haploid cells fuse to form a diploid offspring. Like mitosis, meiosis is preceded by an interphase. During this time, DNA replicates and each chromosome becomes doubled, consisting of 2 identical strands of DNA. Meiosis involves 2 divisions. The first division, meiosis 1, halves the number of chromosomes, and in the second division, meiosis 2, the sister chromatids are split. The end result is 4 haploid cells. Now let's look at each of these divisions in more detail. Meiosis I begins with prophase I. Individual, replicated chromosomes become visible, and each chromosome consists of identical sister chromatids joined at their centromeres. The spindle fibers start to form, and the nuclear envelope disintegrates. In addition, homologous chromosomes line up next to each other, and an event unique to meiosis occurs. During this unique event, called crossing over, two homologous chromosomes, which are lined up next to each other, exchange DNA between adjacent homologous chromatids. After crossing over has occurred, the sister chromatids of one chromosome are no longer identical to one another. In metaphase I, homologous chromosomes line up along the equator of the cell. For each pair of homologs, the orientation on the equator is random. Currently, the large purple chromosome and the small green chromosome are on top. However, the small purple chromosome could just as easily have ended on top. Each different orientation results in gametes with different combinations of parental chromosomes. This process is called independent assortment. Once the chromosomes are lined up, the ends of the spindle fibers attach to each centromere. In anaphase I, the chromosomes move apart from one another along the spindle fiber to the opposite ends of the cell. At this point, each chromosome is still double-stranded and has two sister chromatids. However, we are separating homologous chromosomes into two different cells, so that each new cell will have only one member of the homologous pair and will be haploid. In telophase I, the spindle fiber disintegrates and cytokinesis, cell division, begins. Thus, in meiosis I, which progresses from prophase I to telophase I, two cells are created, and each cell has half the number of chromosomes compared to the original cell. After cytokinesis is complete, a second division, or meiosis II, starts. This division is identical to mitosis, and the resulting cells have the exact same number of chromosomes as the original cell at the end of meiosis I. In prophase II, we now have two cells, each with two chromosomes. In this stage, the spindle fibers again start to form at the poles of the cell. In metaphase II, the chromosomes line up along the equator. This is different from metaphase I, where the homologous chromosomes were lined up. In metaphase II, each cell has only one of each homologous chromosome. In anaphase II, the sister chromatids move away from each other along the spindle fiber, and in telophase II, along with cytokinesis, we see the formation of four genetically different haploid cells.
Okay, let's take a look at spermatogenesis.
Spermatogonia are the cells from which sperm cells arise. The spermatogonia divide by mitosis. One daughter cell remains a spermatogonium, and the other becomes a primary spermatocyte. The primary spermatocyte divides by meiosis to form secondary spermatocytes. Secondary spermatocytes divide again to form spermatids. The spermatids differentiate into sperm cells.
Okay, once the sperm are produced, we have to provide for them a pathway to exit the testis. The efferent ductules lead out of the testis and into the epididymis, which is the site of sperm maturation. This takes about 2 days. The head, the body, and the tail are already formed at this point. Stereocilia are in the pseudostratified columnar epithelia of this structure. They increase the surface area to facilitate absorption of fluid from the lumen of the duct. One of the things also that we have to appreciate about the epididymis is this is where the sperm learn to use their flagella for motility. The vas deferens pass from the epididymis into the abdominal cavity, the ductus, testicular artery, venous plexus, and lymphatic vessels, nerves, and fibrous remains of the process vaginalis form the structure known as the spermatic cord. One of the things that we'll learn to appreciate about the spermatic cord is that the testicular artery and vein act as a heat exchanger to cool the blood entering the testis and keep the temperature, again, 2 to 3 degrees below body temp. The distal end of the ductus deferens is enlarged as the ampulla, where it fuses again with seminal vesicle on its way into the prostatic urethra. The walls of the vas deferens have smooth muscle that engage in peristalsis during ejaculation, again to propel the ejaculate into the female reproductive tract. The ejaculatory duct marks the joining of the vas deferens and seminal vesicle, and it ends at the urethra inside the prostate. What you're looking at here is a superior view of the male reproductive anatomy with the penis bifurcated so that you can see the corpora cavernosa and the spongy urethra and the corpus spongiosum in this portion of the organ. The urethra in the male extends from the urinary bladder to the distal end of the penis and is again a passageway for both urine and semen, with semen again having the right of way. The three parts of the male urethra are the prostatic, which is connected to the bladder and passes through the prostate. Approximately 20 ducts from the prostate that empty into the prostatic urethra in the production of semen. The membranous urethra extends through the perineum and ultimately connects with the spongy urethra, which passes through the penis. There are several mucus-secreting urethral glands that empty into the spongy urethra. The purpose of the bulbourethral gland and these other glands is to prepare the urethra for the passage of the semen by neutralizing the acidic pH left behind often after urination. Sperm do not like a low pH environment and are not viable very long in its presence. The penis is the male copulatory organ. It functions in urination and it serves as a vehicle for injecting sperm into the vagina. Erection is the process of blood via hydrostatic function causing the organ to stiffen in preparation for intromission into the female reproductive tract. 3 columns of erectile tissue engorged with blood, the paired corpora cavernosa on top dorsal side of the penis, and the corpus spongiosum, which of course encircles the spongy urethra. One of the things to appreciate about the corpora cavernosa is that they're expanded at the base, forming a crus, and they're attached by ligaments to the coxal bone. The crura and the bulb are what form the root of the penis, and the glands is an enlargement of the corpus spongiosum. The prepuce covers the glands. Circumcision, remember, is the surgical procedure removing the prepuce for primarily hygienic reasons to avoid irritation, inflammation, infection by the bacteria that like to populate that region. And of course, the external urethral orifice is the exit for semen and urine from the reproductive organ. Okay, the accessory glands that are designed, again, to facilitate semen production. The seminal vesicles empty into the ejaculatory duct and make 60% of the volume of semen. Secretions also contain fibrinogen. The pH is again alkaline so that the sperm don't die. The prostate makes another 30% of the semen. It's a thin, milky secretion with a high pH that contains clotting factors and fibrinolysin. The purpose of the fibrinolysin, of course, is to break apart the fibrin that forms as a temporary plug after the semen enters the female reproductive tract. Essentially a way to lock the gametes into the female reproductive tract so they won't spill out in the reverse direction. The bulbourethral glands make about 5% of the volume of the semen, and they have a mucus secretion that's produced just prior to ejaculation. It helps neutralize the pH of the female vagina. And then, of course, the remainder of the semen comes from the testes in the form of the sperm themselves. The secretions of all three accessory glands plus the sperm are referred to as semen. The urethral mucosa produces mucus. The emission is the discharge of the semen into the prostatic urethra, while ejaculation is the expulsion of semen from the urethra and is carried out, of course, by the smooth muscle contractions we've already become familiar with in the digestive system known as peristalsis. Temporary coagulation occurs as fibrinogen is converted to fibrin, allowing the sperm again passage into the female reproductive tract. Fibrinolysin then breaks up the coagulation, allowing the sperm to swim up the vagina and ultimately find the egg. What we're looking at here is the hormonal axis that's designed to modulate sperm production, and it's against essentially a hormonal communication between the hypothalamus, the pituitary, and of course the target glands, the gonads. So we begin with gonadotropin releasing hormone from the hypothalamus triggering LH and FSH secretion from the anterior pituitary. The luteinizing hormone stimulates testosterone secretion from interstitial cells, which increases the rate of sperm production, among other effects. The follicle stimulating hormone stimulates sustentacular cells of the seminiferous tubules to increase spermatogenesis and to secrete inhibin. Testosterone has a stimulatory effect on the sustentaculars of the seminiferous tubules, as well as the development of the sex organs and the secondary sex characteristics. Testosterone ultimately has a negative feedback on the release of GnRH and FSH and LH, again, in order to modulate sperm production so that it's not constantly at its highest possible rate throughout development. So you can see here the inhibition indicated by the red arrows and, of course, stimulatory effects by the green. Puberty in males marks the beginning of reproductive life. Before birth, the placenta secretes human chorionic gonadotropin, which stimulates secretion of testosterone by the fetal testes. From birth to puberty, no stimulation or secretion of testosterone occurs, and then at puberty, this is the age at which the individual becomes capable of sexual reproduction. Prior to puberty, small amounts of testosterone from the adrenal gland inhibit gonadotropin releasing hormone. At puberty, the pituitary becomes less sensitive to this testosterone inhibition. GnRH production increases, LH and FSH target the testes, and the testosterone production ultimately results in an increase in spermatogenesis and also, of course, the effects of testosterone on the rest of the body. Elevated LH causes the interstitial cells to make the testosterone. The testosterone elevates the rate of spermatogenesis, but also has broader effects. It increases muscle mass, it increases bone density, and it increases hematocrit and metabolic rate, just to name a few of the effects. Most of the testosterone in the body comes from interstitial cells of the testes and tiny amounts from the adrenal glands and the sustentacular cells. And again, the effects are indicated here. Essentially, the male secondary sex characteristics, axial and body hair, deepening of the voice, increased muscle mass and hematocrit and bone density. In addition to this, we have differentiation of the male genitals and reproductive ducts necessary for sperm formation. The descent of the testes is obviously complete by this time. We also see hair on different parts of the body. The skin becomes rougher and coarser. Amount of melanin increases, so the skin will darken. We see increase in production of sebaceous fluid, so the skin becomes oily. We see hypertrophy of the larynx, producing the Adam's apple, and of course the concurrent increase in hematocrit, metabolic rate, muscle mass, and general anabolism. We also experience at the end of puberty the closure of the epiphyseal plates as a result, again, of these hormone shifts. Okay, male sexual behavior is essentially spurred or initiated or enhanced by testosterone production. The male sex act is a complex series of reflexes that results in erection and secretion of mucus into the urethra, emission, and ejaculation. The sensations result in orgasm associated with ejaculation and then resolution, although this doesn't always happen concurrently. Sometimes you can have orgasm without ejaculation, and sometimes you can have ejaculation without orgasm. What you're looking at here are the chemical changes that result eventually in erection. So if we start at the top of the flow chart here, what we'll notice is that action potentials in the parasympathetic neurons cause voltage-gated calcium channels to pop open. Calcium diffuses into the presynaptic terminals, and this initiates acetylcholine release from presynaptic vesicles. Calcium ions also activate nitric oxide, which is a gas, and this promotes the synthesis of, nitric oxide synthase, which promotes nitric oxide synthesis from the amino acid arginine. The acetylcholine binds to the acetylcholine receptors on smooth muscle cells, activating a G protein, which causes relaxation of the smooth muscle cells, and this lets blood into the erectile tissue of the penis, causing an erection. The nitric oxide binds to guanylate cyclase enzymes, and it activates them. The activated enzymes convert GTP to cyclic GMP, the second messenger, and this causes relaxation of the smooth muscle cells and erection in the penis. For those who are interested, what Viagra does is it extends the half-life of cyclic GMP, causing the erection to last longer than it would under normal circumstances, often long after the experience of orgasm and ejaculation occur. Emission is the accumulation of sperm and secretions of the prostate and seminal vesicles in the urethra, and it's controlled by sympathetic centers in the spinal cord, resulting in peristaltic contractions of reproductive ducts. Seminal vesicle and prostate secretions also are accelerated, and we see accumulation in the prostatic urethra, which sends sensory information through the pudendal nerve to the spinal cord, resulting in sympathetic and somatic motor output. The sympathetic output causes constriction of the internal sphincter of the urinary bladder so that the urine is held back while the semen exits via, again, the three portions of the urethra. Somatic motor inputs hit skeletal muscle in the urogenital diaphragm and the base of the penis, causing rhythmic contractions that help force the semen out of the urethra, and this is the act of ejaculation. Now, the next part of our discussion will center on the female reproductive system. What I wanted to do is give you a rundown of the female reproductive organs, and also to let you know that the female reproductive system is considerably more complex than the male reproductive system, because not only does it have to produce the gametes, it also has to provide a home for the developing embryo and fetus. So rundown of the relevant structures include the ovaries, the fallopian or uterine tubes, the uterus, vagina, and the external genitalia, as well as the mammary glands. And we will pick up with that discussion in the next installment of Chapter 28. Okay, I appreciate your attention, and I'll see everybody in class. Thank you.